Hi, everyone. I'm George from the Department of Food Science and Nutrition here at the University of Minnesota, and I'll be doing this presentation with Jim Anderson, and it was put together also with the help of Pravin. We'll be talking on tackling with digestibility, specifically looking at FODMAP, different FODMAP levels in different wheat lines. So wheat is an important crop here in Minnesota and in the US in general, but over the years, we've seen a drastic reduction in the per capita consumption of wheat in the US. This graph here just gives us a glimpse of what has happened in the past 20 years. So from 2000, we've seen a significant decrease in per capita consumption of wheat from 146.3 to 132.1. And we still continue to see this trend go down and down. This doesn't bode well for the wheat industry and also farmers in general. But why is this happening? We think a couple of reasons are contributing to this. One of it is what we think are from fat diets. This is where a large percentage of the population remove starches from their diet, and we know wheat has a significant amount of starch, so that is a no-go there. And then we also have the situation where a lot of people are avoiding gluten and or wheat in general. And some of these diets are paleo, whole, and keto diet. I'm sure some of you know these diets and what they actually do. As far as we are concerned, it's actually we think it's contributing to the reduction in the consumption of wheat. So let's look at the avoidance of gluten and or wheat. This is a major factor, and it is basically due to gluten. A lot of people who react to gluten, especially people who have celiac disease. We know celiac disease is an immune disease in which people just cannot eat gluten. It is found in wheat, rye, and barley, so it is very necessary. This occurs in just about 1% of the population, but that 1% has really still made the consumption of, have contributed to that decline in the consumption of wheat. There's also wheat allergy, and wheat allergy occurs when our bodies produces antibodies to proteins that are found in wheat. And then you also have non-allergy, non-celiac wheat sensitivity, which is when people react in a way, and people react negatively to wheat, but not necessarily because they have celiac disease. So this pictures here are just showing the normal lining of the small intestines for a normal person, and when you really have celiac disease, so it can be a serious disease or a serious condition. So if it is not gluten, what is it per se, right? We think that this decline in wheat consumption can also come from FODMAPs and ATIs. So FODMAPs are fermentable oligosaccharides, monosaccharides, and polyols, basically made up of short-chain fructooligosaccharides and also amylase trypsin inhibitors. These are a small group of 17 proteins that we know also can cause an immune response. This graph here shows the levels of FODMAPs in different foods. This graph here shows mostly FODMAP levels in baked foods, and then it can also be found in vegetables, fruits, onions, watermelons, and all that. But basically, you can see that there's a cutoff point. Now, most people can tolerate FODMAPs when it is less than 0.3 grams per serving, and this per serving is probably just 2 slices of bread, which is just 72 grams. So you are looking at a very small amount of these FODMAPs that can really cause a lot of discomfort in people. So to tackle this issue, the Minnesota Department of Agriculture funded a project hosted by the University of Minnesota and AGRI. And of course, we've been working on this with the Minnesota Wheat Research and Promotion Council, and also with Backwind Foods, which is a private food company. And we agree that this is a serious issue that has to be dealt with. Now, what are the long-term goals? Reduce the discomfort resulting from the consumption of wheat-based foods. This will inevitably result in improving the health of consumers and, of course, increase the profitability of wheat farmers, because we believe this will increase the demand for wheat. So it's a win-win situation. What are the specific objectives? Characterize variation and identify genetic markers for FODMAPs and ATI in ancient, heritage, and modern wheat varieties grown in different parts of Minnesota. We also will be exploring the use of fermentation as a technique to reduce levels of FODMAPs and ATI in wheat products. Working with AGRI, we will want to establish pathways to implement the research outcome to the food industry, to the wheat industry, and one of which is through a conference like this. So I'll hand over to Jim to talk about the materials and the methods and how we made this work. So Jim.
Okay, thank you, George. So we started out with a panel of 220 different wheat lines. Most of these are varieties. So we're trying to get a very diverse group of materials, and I'll show the details on these in the next slide. So this panel, it really represents the history of the U of M breeding program, which dates back to 1895, and also some prominent varieties from our regional breeding programs as well. Then we did a DNA fingerprinting exercise on all of them. We used a technique called genotyping by sequencing. So it's modern DNA fingerprinting technology. It costs about $10 per sample, which is a fraction of the cost of what it used to be. So a study like this wouldn't have even been possible 15 years ago because of that. And then George's lab has determined the FODMAPs on all of these materials, and the ATIs are currently in process. And then we put all of this together by doing what we call association mapping to try to identify DNA markers associated with the different levels of FODMAPs and ATI activities in these wheat lines. So here's the distribution of these wheat lines. So the first group here we're calling the heritage varieties. So there's 46 of them. So we've got a pretty good group. We define those as being developed before 1970. 1970 is when the first semi-dwarf wheat lines were grown in Minnesota. So I kind of made that arbitrary delineation there. And one thing we wanted to look at in the study was, our FODMAP or ATI levels increasing over time? Is it something that we maybe inadvertently have bred for during the breeding process? Most of the panel is modern wheat, so a large diversity of materials there. And then these others are kind of outliers, just to see what sort of variation we see there. So we have some durums, einkorn, that's one of the progenitors of wheat, emmer, also one of the progenitors of wheat. And these synthetic hexaploids, they're made by crossing emmers or durums with the third progenitor of wheat. So a lot of genetic diversity here. When we grow them out in the field, both at St. Paul and Crookston, we have to treat them with fungicide, or else the fungal pathogens will reduce the yield and cause shriveled grain. So there's a lot of diversity evident in these materials when we get them out into the field. So based on our genetic fingerprinting analysis, we can make a scatter plot like this. So every dot on here is the name of one of these 220 lines where we did the DNA fingerprinting. And this particular analysis explains about 20% of the total diversity present in these materials, and they can be broadly classified as bred wheats over here on the left, the durum and synthetic hexaploids, and then those emmers or the ancient durums in the lower right-hand corner. So this is kind of what we were expecting. And then to get a little bit more definition within that bread wheat group, we redid this analysis showing just the bread wheat line. So we eliminated those synthetic hexaploids and the progenitor lines. And what this amounts to then is we can sort of separate out these older heritage lines and founder lines. So these are kind of the foundation, if you will, of our breeding program going back into the early 1900s. They roughly cluster in one area. And then we have Chinese, Mexican, and South American lines that were used as parents in the breeding program at various stages. They roughly clustered together. And then we have all of the modern lines, those that were developed after 1970. So what this analysis shows, again, is that we have a good amount of genetic diversity in these materials, which is what we were striving for.
So this slide here shows how we actually do determine the FODMAPs and the fructans in the samples. We use the HPLC, and this is a typical graph we get. So you have all the sugars that appear on the graph, and they are identified with standards. So we can classify them into the different sugars, and also we have the fructans in here. Now, based on a graph like this, we were able to quantify how much of these FODMAPs were, or fructans were in the samples. This is showing 196 samples that we ran in Crookston. As I said, we have a complete set now, which we can present later on, but you can see a wide variation in some of the monosaccharides in these different samples. So you can see here the total fructans. You can see here the FODMAPs with excess fructose, because fructose is very important in contributing to this issue of FODMAPs actually irritating our intestinal walls. And then you will have the fructans with less than DP10, which are called fructooligosaccharides. Now, if you look at this graph here, you could see there are very wide variations in the samples, even though they look very small after about 1.6%. If you go back to the fact that we need just more than 0.3 grams per serving, these values are still significant. So if we can do some breeding to really reduce the levels of breed for wheat samples that are really low in some of these FODMAPs or fructans, that would really be very, very important. So James would continue on the slide.
Okay, so this is the genetic analysis part of it. So what we do with those DNA fingerprints that we collected, and I showed earlier how those could describe the genetic variation among those wheat varieties. Now we take those DNA fingerprints, compare it to their level of FODMAP data, and try to find out or identify specific regions of the genome or even genes that are responsible for that FODMAP variation. So on the x-axis here, we have the 21 chromosomes of wheat, and there's about 4,000 DNA markers spread along these chromosomes. And the vertical line, or the vertical distance here, indicates the significance of a particular DNA marker with FODMAP levels. So anything above this horizontal green line, it's kind of a statistically significant association. So we would say that there's some gene affecting FODMAPs here on this chromosome, 2 more over here. Now this is a preliminary result, but it does show us that this is a trait that's probably under complex genetic control. We were hoping to get lucky and maybe find just 1 or 2 genes controlling the trait. That would make it easier and faster to breed for, to reduce FODMAP levels. But it looks like it's going to be a complex trait based on this early analysis. So to summarize our preliminary findings, so we have a genetically diverse set of materials. We have wide differences both in FODMAPs and fructans that we've seen so far. One thing we were looking for was that a trend, so our FODMAPs increasing with the more modern wheat varieties, and we have not seen that so far. So that's good news that we haven't inadvertently been increasing FODMAP levels through our breeding. And we've also discovered that there's no genomic region that seems to be responsible for a large portion of the variation. This will make the breeding a little bit more difficult, but still not impossible. So I'd like to end by acknowledging several other researchers in our lab that are very critical to conducting the research, and once again acknowledging our funding source, the Minnesota Department of Agriculture. And we'd be happy to take any questions.
Yep.
Okay, the question for you, it is the expectation that reducing FODMAPs and fructans will have no negative effect on end-use properties?
George, can
thank
you
you very
answer
much
that?
for that question. Yes, so thank you for that question. Yes, that is the expectation. We don't want to change any of the end-use characteristics significantly, and that will be something that we will be really keeping tabs on. Yeah, we don't want to do that. We just want to have wheat varieties with very low FODMAP, so would improve digestibility issues.
Okay, we got one more question. Why do you think FODMAPs are more of an issue now?
Okay, so I guess it's me again. So, I mean, over the years from the presentation, you've seen that we said consumption of wheat is going down. There's all the issues with gluten, but we think that it is more than gluten. Why now is because there's been a lot of research that has come up to show that consumption of FODMAPs actually causes some of these issues. And with advancements in kind of analysis techniques, now we use the high performance anionic exchange chromatography that is able to let us really quantify these FODMAPs in very good detail. We think it will be a very good time to kind of start screening these samples to make sure that, as Jim says, we are not innovatively breeding for high FODMAPs.
Thank you, George. I got a kind of general broad question here. So can you say anything about heritage or ancient grains?
I think that goes to Jim.
Yeah, no, not at this time. So that's one thing I was really looking for when we started getting the FODMAP data in, and I'm really not seeing any patterns there. Some of the synthetic hexaploids were kind of high, some were on the low end. So right now we really don't see a strong pattern emerging there. Now keep in mind, all we've looked at so far are the FODMAPs. We still have the ATIs to look at, and based on the literature that I'm familiar with in this area, I think the ATIs are maybe going to explain more of some of these issues than the FODMAPs actually do. And then we also have the processing, so looking at different fermentation.
Yeah.
I think that's all we've got for you guys. We appreciate your guys' time. Thank you.
All right. Thank you.
Thank you.
All right, thank you, doctors.